So there was a neat article that came out about the mechanism of LDL getting into the artery wall. As I mentioned in the video, I did a video on it. Sorry about that. I did a video on it, and here's the image. Now, what's interesting, I warned folks. I said, authors of studies tend to overstate. They're like everybody else. They're humans, just like everybody else. They tend to market their work, and they tend to overstate the importance and significance. This author, for example, said this totally trashes the inflammation process. This thought about holes in the endothelium. Many of us have used that term mostly to help folks understand what's going on. And I'm personally very guilty of that as well. It's hard enough. This is a complicated enough subject. You do anything you can, and sometimes you take shortcuts to try to simplify it for the lay public, because the lay public needs to know some of the science in order to stop the process internally. Anyway, I'm going on too long about it. Here, let me just go on and talk about one of the comments that somebody made. Patrick Neighbors. Hi, Dr. Ford. This study, if true, pretty much wrecks the inflammation lesions model for causing disease. I've watched your videos. Thanks very much, and have adopted low carb, low blood glucose, etc., etc. Now, my response was, as I said, I don't think it changes much in terms of prevention at this time. It does nothing to the concept of inflammation, low carbs, etc. But if the damage is associated with a specific metabolic pathway, instead of totally denuding the endothelial cells, this is a big opportunity for pharmaceutical research. That was the point, and it's still the point. And then Eldine jumped in and tried to help. Now, if you're listening to all this and saying, wait a minute, what on earth is he talking about? Let's go back and just do a couple of basics real quick. Most people think that when the arteries get plaque in them, it's like hair lining the drain of your shower or tub. It lines the inside, and it just continues to line it deeper and deeper, or cover more and more of the lumen, or the hole, to where you can't get water out the tub anymore, or you can't get blood through your artery. That's not what happens. What happens is there are three layers. There are two critical layers of the artery. The intima layer, or endothelium, it's the smooth lining. And then the muscle layer. That supports that thin single cell endothelium. So when LDL, bad cholesterol, low density lipoprotein, when you get too much of it, it can start going through this intima wall, and it starts to pile up in the media, the muscular wall. And in fact, it piles up in between the two. As you see here, this is a progression of arterial plaque. As it gets deeper or thicker and thicker, you still have that smooth endothelium, but you have this growing plaque. And the immune system says, no, that's not right. I need to attack that, get that out of there. Pretty soon it causes what we call hot plaque. That hot plaque is liquid. If it breaks back through the endothelium and touches the bloodstream, it can cause a clot. Now that sounds very good theoretically, right? Well, again, let's just look at what actually happened. This is an actual artery. This is the endothelium. There's a tiny single layer here. This red stuff, this thick red layer, is the media. So the intima and the media. This big gray thing with these layers is plaque. And that's, again, simply LDL, low density lipoprotein, which is piled up. Now this is a waxy substance, and that's not really going to stop the blood. It's not going to create a problem with with a clot. But here, look at this section right here. As you get deeper into this section and blow it up further, what you see is it's actually liquid. And this is a section of liquid or hot plaque. The immune system has gone in, trying to dissolve that waxy plaque substance, and it does what it always does. It releases enzymes. Those enzymes have dissolved that plaque. But again, the enzymes and some of the other particles in there can create a clot if it touches the blood. Well, let's go a little bit deeper. This is another view of again, the last one was an actual artery. This is an actual artery as well. It's a different type of staining, so you don't see the red. But this is the intima layer, that simple layer right there. The medium or muscle layer is more of a clear layer over here. This area was an area of hot plaque. As you see now, it's black and shiny. It is a clot. What happened was that hot plaque did break through in these cracks right here through the intima. It touched the bloodstream. It formed a clot. This is a part of the clot, but the major part of the clot flowed on downhill, downstream, went to the heart, and killed the patient. So when these hot plaques release, if they cause a clot, if the clot's big enough and it goes to the heart, it causes a heart attack. If it's big enough and it goes to the brain, it causes stroke. Now let's go back, and the original question here was, did this actually wreck the inflammation mechanism, inflammation theory? And again, the answer is no. A little bit of hyperbole there, a little bit of overstatement of what's going on. What I'm going to do is go back and show you an article from the British medical journals, postgraduate medical journals. It's a lit review from my friends, Brad Bale, Amy Doneen, and David Vigorist. And this one has to do with periodontal pathogens and contribution to atherosclerosis. I'm not going to get too deep into the periodontal piece. I've covered that periodontal piece many times. I've had Doug on here many times. But here is here's the mechanism. And again, this mechanism was described in that article, journal article. I'll provide a copy of it or a link to it in the description below. Basically, you're looking at what's called the atherogenic triad. And the atherogenic triad does include, people say, well, it's never LDL. Having an elevated LDL is an issue. Increased serum lipoprotein concentration is one of the atherogenic triad. That alone, though, usually does not cause significant problems. It's when you start getting the next issue, dysfunction and permeability of endothelium, allowing adhesion and penetration of monocytes along with lipoproteins. And in fact, beforehand, that was the significance of this last study. The dysfunction and permeability of the endothelium is also allowing transfer of the LDL, oxidized LDL, from the bloodstream through the intima layer. That was what this article was about. And he did some great work with some molecular biology showing that that was exactly what happened. So we didn't really have to worry so much about denuding of that endothelium, wiping out the endothelium, holes in it, things like that. But we did still have to worry about, just as we knew before, increased permeability of that endothelium. Now, to go on and just cover a couple of other things in this article, what was the third component? So the first component of the triad was some increase in the LDL. The second component was dysfunction and permeability of that endothelium layer. The third component was binding of ApoB and proteoglycans generated by the migratory smooth muscle cells. So again, once you start getting that transfer of the LDL through the intima and into the media site, the immune system starts attacking it. It starts having an impact on the smooth muscle cells. The immune cells start releasing enzymes, et cetera, and here we go down that path. This video is getting a little bit long. I'm going to go ahead and finish it out. I think maybe I'll come back and go into more of the molecular mechanism regarding periodontal pathogens and their impact a little bit later. If you've made it this far, thank you again for your interest. Thanks, and if you hit that subscribe or like button, it makes a big difference. An even bigger difference happens when you share. You can share on Facebook, Instagram, LinkedIn, Twitter, Pinterest. When you do that, it makes a big difference in terms of the algorithm. It sends this to other people, realizing that humans think this is interesting information and helpful. Thank you again.